Welcome, IWMF volunteer leaders and international affiliates from around the globe. I am Lisa Wise, IWMF Vice Chair of Information and Support, and I want to offer wholehearted thanks to our worldwide volunteers for all that they do to support and educate their fellow WMers. Today's call has participants from the United Kingdom, New Zealand, Canada, Australia, Finland, and the US. We are all in different time zones across the world, demonstrating that it is always the right time to gather together, get educated, stay informed, and support one another every step of the way. We are all in this together. Huge thanks to Michelle Postic, IWMF Manager of Information and Support, my fabulous co-host today, and my WM partner in crime, and a veritable fountain of resources. Thank you, Shelley. I am simply thrilled to introduce today's most exciting program, one that we've all been waiting eagerly to hear. We'll be learning from a world expert about a hot topic that is top of mind these days, the COVID vaccine, WM, the FDA-approved booster. So much to unpack. What a sheer honor to extend my warmest welcome and express my deepest respect and admiration to Dr. Larry Saltzman, Executive Research Director of the Leukemia and Lymphoma Society. Thank you, Dr. Saltzman, for graciously agreeing to present on the Michael J. Garil Patient Data Collaborative. We cannot wait to hear you explain how the LLS Registry has been used for COVID vaccine insights. Your tireless advocacy and personal efforts are legendary. As a blood cancer patient yourself, we know that you have skin and cells in the game, and that it's not been an easy ride, but you are simply unstoppable. So in this marathon of life, we all benefit from your grit, and we are inspired by your resilience. All of your efforts at LLS are achieved with deepest commitment, dedication, and energy, and with the fullest heart. And for that, we thank you, thank you, thank you. Thank you also to LLS, a cherished partner of the IWMF, for all of their invaluable research, educational programming, and exceptional support services. I have to admit that I'm not a science maven, so it was hard reading and getting through all of this material, and that's why I've been looking forward to this. But I can easily understand a few things about this incredibly impressive research effort, and that is, this was the largest COVID-19 vaccine study to date to report detailed findings by blood cancer and treatment types. Dr. Saltzman recently presented at a pivotal CDC meeting to discuss booster vaccines for the immunocompromised. Thank you for being that voice. And this LLS research was a catalyst for the FDA's approval of a third vaccine. They found that non-Hodgkin lymphoma patients are less likely to have detectable antibodies. Many of us on this call are living with that reality firsthand. And they found that multiple myeloma patients had higher rates of antibodies compared to other blood cancers. I don't want WMers to be jealous. I want us to celebrate for multiple myeloma patients. We're coveting their response. LLS experts say that vaccination offers protection to most blood cancer patients. Again, we're a little jealous, and should be combined with prevention precautions for best protection. So welcome, Dr. Saltzman. We owe LLS a debt of gratitude, and thank you so much for taking the time to be here to teach us today. We all can't wait for me to turn off my microphone so we can listen to you. Please take it away, Dr. Saltzman. Thank you.
Thank you, Lisa. Pleasure to be here, and I hope you can all hear me okay. Somebody give me a thumbs up. Good. I am going to use the power of Zoom to share my screen, and I have put up a PowerPoint. We can see this. Yes, say yes. Okay, Lisa's thumb up. All right. Thank you again. And the purpose of this webinar, if you will, is to provide information regarding the progress of our LLS National Patient Registry, to present findings from our initial publication, which was brought out in the journal Cancer Cell about 2 weeks ago, and to answer your frequently asked questions as presented and probably more. I am a family physician. I'm not in clinical practice any longer. I founded a health IT company, which I ran for 15 years regarding big data, claim submission, eligibility verification, and the like. I am, as Lisa said, a chronic lymphocytic leukemia patient. I was diagnosed in 2010. I have a poor prognosis. Frankly, was given an 8-year life prognosis in 2010. Well, I'm still here today. My most recent treatment was CAR T therapy in December of 2019. And I am the principal investigator for the LLS National Patient Registry. And let's do this again. Sorry. My treatments have included things that are familiar probably to most of you, bendamustine and rituximab, ibrutinib. I had to have my right colon removed for a lymphoma obstruction in 2015. I was on the original venetoclax clinical trial and continued with that for 3 years in conjunction with ibrutinib. I've had multiple chemotherapies, CAR T therapy, and I am on monthly IVIG. So a mixture of just about everything. So we're all friends here, and want you to know that. Now, this is an observational study, meaning it's not a means to evaluate new treatments or medications, and that's important. We are looking at booster monitoring, and I'll get to that. It is important to note that LLS is not a medical provider. We receive many questions about should I, shouldn't I, and we really can't answer those. We can collect patient-reported outcomes via surveys, and I view this as akin to something called the Framingham Cardiac Study, which was launched in the 60s in a town of Framingham, Massachusetts, where they've been studying their population ever since. And it was through that observational longitudinal study that we have come to learn that smoking and high cholesterol and high blood pressure really do cause heart disease. It came out of that. And so we hope our study will be able to observe patterns in the future. Our technology partner is a company called Citizen, spelled with 2 I's, and we get questions about this because they want to know if this is a real company or not, and it is. It was founded by a fellow whose sister passed away of breast cancer, and they had trouble getting her medical records from place to place, and this is what they do. They offer for us HIPAA-compliant medical record collection, and that's part of our registry. In getting the records, there's no work on the medical provider's part. We request the records, we collect the records. There's nothing a doctor's office needs to do. The record requests are updated on a periodic basis, so we don't have to request records every time a patient sees their clinician. We do collect patient-reported outcomes via a survey. We have invitations for outgoing research. If there is research, the PHI will be stripped from all the records, so they will just be pure treatments and the like. LLS approved researchers will study the records and results. And if we have an outside party who wants to review our data, the participants will be asked for consent. So nothing will get done without a person's knowledge of what we're doing with this. Now, the basis of this study is that blood cancer patients are among those who are at increased risk of developing more severe illness from the virus that causes COVID-19. This has been reported, and it includes a higher risk for severe infections and death. The initial COVID vaccine trials did not include blood cancer patients or virtually any cancer patients at all, leaving us without important information about how well the COVID vaccines work for them. The vaccines may provide less protection to people with some types of blood cancer or blood cancer treatments like chemotherapy. And this is what we're attempting to study. Our registry itself was launched February 22nd. That was the initial launch of will you share medical records with us? The COVID antibody response study was launched March 18th. So really not quite 5 months ago. This has been an incredible, intensive effort. We've had 10,000 patients, over 10,000 register as of July 31st. Actually, it says 3,800 patients, but now we're close to 4,000 patients have submitted blood samples for SARS-CoV-2 antibodies. We did publish a paper in Cancer Cell based on the analysis of the first 1,400 lab samples as of May 12th. So yes, there's a time between then and now. And this is the largest COVID vaccine response blood cancer study in the United States. Surveys were collected from participants that formed the initial study interpretations, and medical records for specific treatments related to COVID or specific blood cancer treatments will be processed in the coming months, so we'll be able to report a deeper dive. The lab tests that we're asking for are a Roche assay. This is important because there are many blood tests out there for COVID antibodies, and frankly, we need to be able to compare apples with apples. The Roche study is the most sensitive. It's being used in the UK and other areas, and it is the lab that LabCorp is using, and they were one of the first to establish this, and that is why we're focused on our participants going to a LabCorp facility. We check for the nucleocapsid antibody, which diagnoses a COVID-19 infection. This will be positive whether a patient reports it or not. So it's interesting. It's reported as positive or negative. And the reason we need to check this is because when we look for the semi-quantitative spike antibodies, meaning the test that measures the body's response to the vaccine, it also measures the body's response to a natural infection. So if we get this paired sample from a participant and we see that their nucleocapsid test is positive and they also have an antibody response, we're basically going to assume that was from the infection and not from the vaccine, which is important for our study. We have to be very clear on what we're reporting on. The semi-quantitative spike antibody test is a combination of multiple antibodies. It's IgA, IgM, and IgG. And we know from an infection or a vaccine that IgM is the first antibody produced and it follows up with IgG. Again, we are not doing specific blood tests for each one of these. Other laboratories do measure these, and actually LabCorp has a test for IgG only, but we want the collection of this combination for our study. It's reported as a numeric value, and it's limited to the test sensitivity, and I'll go over those values in a while here. So here's some perspectives on the results and the numbers. We are measuring semi-quantitative spike antibody. This includes, again, a measure of these three antibodies, IgA, IgM, and IgG, and simply stated, a value that is less than 0.79 equates to no response. And most of these are reported as less than 0.40. Now, I want to give a disclaimer about this because people get kind of wigged out when they see this number. They say, well, I don't have any response. And the truth is the sensitivity of the test cannot measure less than 0.4. People may have antibodies, but it's just not measurable by the sensitivity of the test. And so this is the cutoff. If a value is greater than 0.8, it indicates, again, within the sensitivity of the test, that there was a response to the vaccine. Now, LabCorp threw a little loop in our results in that somewhere in the middle of May, Roche assay was able to increase their positive sensitivity rate, which was reported before May as greater than 250, and is now reported as greater than 2,500. So the result range has expanded. Having said that, if somebody had a previous response of, say, 0.8 to 250, that response is apples to apples with the new top number. So if somebody's number was 200 before and 200 today, those line up. We're not going to take the new response and multiply it by 10 or divide it by a factor of 10. In other words, all they've done is increase the sensitivity to now report that if the number was greater than 250 before May, it could now be 1,500. I mean, those are all greater than 250. So it's a little confusing, but a previous or current response of up to 250 remains the same. Okay, so we'll probably get questions about that, but that's the answer. Now, how do our responses compare to the normal healthy population? We get this a lot. The vaccine trials were based when the lab values reported of 0 to greater than 250. That was before they changed the sensitivity test. There are no official reports of what number is protective. And because of this, the CDC in general has recommended that the average general population not get these antibody tests because they cannot correlate a test to who will or will not become infected with COVID. They do know that two values pop out. 130 is the value that the Red Cross blood banks use to decide if a plasma sample contains enough COVID-19 antibodies to be used for plasma treatments. It is also the ballpark number that research papers have associated with an adequate healthy response. So if your number comes in at 130 or above, you pretty much match up to a normal population. A result of greater than 200 has been said to imply a full response. So whether your number is 200 or greater than 2,500, there is no data as to which number is better. All we know is that you have a response to the vaccine, and we still tell our population and our participants to keep your guard up. Don't relax just because of this number. Okay. Now, the results presented to you following this slide will be generalized on the types of blood cancers and treatments. This presentation, or most of our results, cannot touch on any personal results. So that's a little bit of a challenge. Individual results should be shared with your provider, and the reported side effects of our participants from the vaccine are no different than the side effect profiles of the vaccine trials. We haven't seen anything different, and therefore we encourage everyone to get vaccinated and act unvaccinated. That's our motto at LLS. As I said, the results were published in Cancer Cell. You can find that article, data reviewed and collected as of May 21st. And since May 12th, as we analyze the data, the trends I'm going to show you, we are finding are really not changing. The numbers may change just a smidgen, but the trends are not changing. So here we go. In the big picture, we reviewed 1,445 results, and 75% of blood cancer patients have an adequate antibody response. And the additional data shows these trends are persisting. For the 25% who are seronegative, meaning that they have a number of less than 0.79, these percentages vary by the blood cancer type and or the treatment, and I'll give you a sense of that. Our findings were statistically significant. We ran them by statisticians, and in general are in line with studies published in the UK and Israel, as well as smaller studies in the US. The following are results include all participants, whether recently treated or not. And after these general results, I'll focus a bit on Waldenstroms, which I know you're all, looking for. So these are pie charts, and the pie charts are red if there was a response, meaning, as I said, the response is, greater than 1, let's say, and gray means there was no measurable response. So in the myeloma space, it's really good. If a person was diagnosed with smoldering myeloma, 100% of those people responded to the vaccine. Even people with multiple myeloma, 95% of those folks responded to the vaccine. So these are, very comforting numbers. If you had a myelodysplastic or myeloproliferative neoplasm, again, 97% of these participants responded to the vaccine, a very good number. Okay, as we get into the myeloid leukemias, again, it looks very good. Chronic myeloid leukemia, 97% responded. And this is a combination of people who are on treatment like, Gleevec or Sprycel, or now on holiday. And for the acute myeloid leukemias, AMLs, again, 91% responded, excuse me. And part of the non-responders very likely were because they're on bone marrow transplant or some treatment that affected their immune side of their house. Okay, the lymphoma results, this is a lot of pie charts, but we'll go through them. These are lymphomas, and you can see I've kind of measured them from best to not so good. So Hodgkin's lymphoma patients do very well, 98% respond. We think that's because many Hodgkin patients are diagnosed when they're young. They've had their treatments when they're young. Our registry participants skew to an older population. So these are people who were treated many years ago and they're just fine. They're like normal, healthy individuals. And you can see the results for T-cell lymphomas, non-Hodgkin's lymphomas, not specified, marginal zone, diffuse large B-cell lymphoma is in its own category. You can see a little worse, 79% respond and the rest don't. And mantle cell lymphoma has a problem. 44% of people respond, more than 50% of people did not respond. Okay. Now with the leukemias, I'm in this group. So ALL had a pretty good response. Follicular lymphoma, not so good. Waldenstroms, not bad. 74% of Waldenstroms patients respond to the vaccine. 25% of you or so did not. And I'll go through the medications and maybe there's some explanations. And in the CLL group, again, 64% responded and 36% did not. So not as good with CLL, not quite as good. Now, this is a table, a lot of data here, I'm sorry, a lot of data about the response among CLL patients. And this is telling for other groups. If you look at the top line among treatment within the past 2 years, now we did not break this down to the past month, 6 months, a year, 2 years. Our survey only asked people, have you been on these treatments within the last 2 years? We will break this down when we crack the medical records and we'll know exactly what treatments are being given when based on the vaccine dates. Having said that, the top line, which says none, mean that 17% of seronegative patients, in other words, CLL patients who did not respond, are not on any treatment. And we feel that this is because of their disease, which is a disease of B lymphocytes. And B lymphocytes create antibodies. And so in CLL patients, just because of their disease, their B cells don't act normally. And therefore, they did not create anti-antibodies. The next one down is really telling. It's like a swing of a pendulum. For patients who have been on obinutuzumab, that's a big word, sorry, 90% of these people did not respond to the vaccine. That's a big number. And if you look at the next one, rituximab and acalabrutinib, if they're on that combination, 85% did not respond. Now, we know a lot of Waldenstroms patients take ibrutinib, and if you look down at the bottom, if you're just taking ibrutinib alone, 48% of patients did not respond. So if you think about this as it relates to Waldenstroms, the Waldenstroms group in general may be on wait and watch, or they may not be on treatment. But for those who are on ibrutinib, it skews the numbers for Waldenstroms for those 25% who may have not responded. So again, what you're looking at is a table of treatments and combinations of treatments. And for those who did not respond, these are the treatments that they're on. Interestingly enough, we don't quite understand why people on IVIG would not respond if they're not on any other treatments. And it's probably because of their CLL. Frankly, we're seeing now people who get IVIG who have a serology test and come up positive to the nucleocapsid test. And the reason for that is we've been told that as of the month of May, because IVIG is pooled serum, we're now seeing COVID antibodies in the IVIG prep. And it may be that when we do a blood test for somebody who just had IVIG, I just had my infusion yesterday, I may show a nucleocapsid antibody, even though I know I haven't had COVID. And if I do my serology, I may show a IgM or a semi-quantitative boost, not from my vaccine, but from the IVIG. So this is something we have to look into. I know it's complicated, but just want to bring that up. So now what about Waldenstroms? Okay, here's our data as of August 13th. So this is just like this week. I did this. So of our total participants in the registry of 8,878, 402 of them have identified themselves as having Waldenstroms. That means 5% of total registry participants have Waldenstroms. This compares to a total of 2,700 people who have CLL. That's our biggest group. Other leukemias are 816, other lymphomas are 2,803, and myelomas have a pretty good representation of 1,100 or so. Of the 402 participants who signed up for the registry, we have blood serology on 152 Waldenstroms patients, and this serology is noted a minimum of 2 weeks after vaccination. Now, why the other 250 Waldenstroms patients didn't have their blood drawn, I can't tell you, because this is open to everybody and it's not that hard. Now, some people don't live near a LabCorp site, that is true. And if a Waldenstroms patient lives in Hawaii, there are no LabCorp sites in Hawaii. There are no LabCorp sites in Alaska. And so this is lower 48. And some people live miles away from a LabCorp site and they don't want to make the drive. So that explains some of it. And but we'll leave it at that. So we have data on 152 people. And here's a deeper dive into those who showed no antibody response or a maximum response. Now, we've defined no antibody response as less than 0.79, and we've defined a response like a normal healthy person at more than 130, as I've said before. And the treatments listed per our survey are within the last 2 years. We don't know when within the last 2 years they were. So you can look at the sample sizes, and we have 59 people who are on no therapy. And you can see of the 59 people who are on no therapy, only 3% of them did not have an antibody response. So if you have Waldenstroms and you're not on any therapy, my advice to you is the vaccine is working. Keep your masks on. And don't go dining indoors. That's okay. That's my own, editorial comment. But you can see that, if the highest group that did not respond are the people who are on Rituxan and ibrutinib. If you're on that combination, 63% of these 11 patients, again, it's a very small number, did not respond. But I think that is able to be extrapolated to a group at large. So you can look at these and see where you are. Again, this is a very limited sample, but if you're taking IVIG and that's all you've reported, right, then essentially nobody, didn't respond. 66% of people are above that 130. The rest of the people are between 1 and 130. Everybody responded if you're on no treatment except IVIG. So I'm sure we'll get some questions about this, but this is the current study for your constituents. Okay, I'll let that settle in for a couple of seconds, and here we go. So I want to talk about where we're going after this. Now, LLS is very sensitive to the fact that there are 3 functions in the body to fight infections. One is an antibody response, and that's what we've been talking about. The second is a T-cell response. These are killer T-cells that are actually the first to respond and go kill the infection. And the third is cellular immunity. Now, cellular immunity would be the body recognizing something as it's seen before, as in my arm got really sore after my recent 3rd vaccine, and I had a little low-grade fever, and I attribute that to cellular immunity. But we cannot measure that. Now, T cells we can measure. However, for COVID vaccine response, there is no commercial T-cell test that can measure this. So LLS has entered into a partnership with a company called Adaptive in Seattle, who does genetic testing for clones of different cancers. As an example, my cancer, my CLL, is followed by MRD testing, which stands for measurable residual disease or minimal residual disease. And every few months, I have my blood sent up to Adaptive, and they do what's called a clonoSEQ test. And they tell me how many needles in a haystack I have, meaning how many cancer cells are in my blood to a sensitivity of 1 in a million. Now, Adaptive has a research test to look at T-cell response to the COVID vaccine. But this is not a commercial test. And it's a research study where all the tests at the lab are de-identified. They have no idea who these people are. They just know that's a study number. And therefore, personal results cannot be returned. And this is a problem. People are who we've invited, 1,100 people we invited for T-cell studies. Most of the people were seronegative, meaning they didn't have an antibody response, and we want to know if they had a T-cell response, because this would be important to know. And we are not going to be able to give personal results. We will report these results based on blood cancer types and treatment types. So if, just like in the previous slide in a graph, if we report on a treatment type, we're going to find out what or where people lie within that group, but we can't give personal results. We are doing a 3 and 6 month plus serology follow-up to determine changes over time. We are looking for people who've had CAR T therapy like myself to see what happens during CAR T therapy. And we have started a booster observational study, which right now is a little bit interesting because we just learned today or yesterday that as of September 20th, all Americans will be eligible for a 3rd shot. So really our study is going to turn not a booster study per se, but following serology after multiple vaccinations. Now, I want to tell you that LLS itself is not offering any boosters. You have to get them from where you were. We are not a medical provider or an academic medical center. We can't recommend or comment on individuals' booster questions. We are encouraging studies on these additional vaccines to assure safety in blood cancer patients. We are funding a study at the Montefiore Medical Center, and they are recruiting, and we're looking for more centers. We're closely following the experience in foreign countries where boosters are being investigated in the UK, or being administered in Israel. Israel is now vaccinating with third vaccines anybody over the age of 50. LLS will monitor antibody responses in participants who notify us that they're receiving such a vaccine. The formal booster trial is now, frankly, as of today, kind of a moot point because it's going to turn into an observational trial of multiple vaccines. Okay, FAQs. So I'm going to run through what I've been asked to answer, and then I'll stop and you can ask me questions. Is this study from LLS open outside of the USA? The answer is no. If lab tests are done at LabCorp, how long does it take to process? It's about 3 to 5 business days, sometimes shorter, sometimes longer. The lab slip are sent from our citizen partner or from our support center. And this is our email address, pact@lls.org, where you can write to us. LabCorp will not accept a lab slip displayed on a mobile device. They have to have a printed document. So this is a hassle, but that's the way it is. If LabCorp is too far away or convenient, can our participants go to another lab? As I answered, we want to compare apples with apples, so the answer is no. Now, having said that, there are some clinics that have pickup arrangements with LabCorp. So if the clinic draws the blood and puts it in an envelope with our lab slip, and the LabCorp courier picks up that blood and takes it to a LabCorp site, that's okay. We can do that. The lab results are seen in the LabCorp portal, if you have one, or by calling the LabCorp 800 service number, 800-282-7300, or we are trying our best to post the results in the citizen site for those participants who have signed up, for a citizen account. Do we send the lab slips to a person's doctor? The answer is no, we just can't handle this. So patients can get their results and then bring them to their doctors. Okay, what do the results mean? Okay, we've gone over this a little bit. The nucleocapsid test denotes whether the patient has or has had a COVID natural infection. And the spike measures the antibody response either to vaccine or to this infection. We explained what the 0.8 cutoff means. If it's lower than that, no response. If it's higher than that, you've had a response. Again, the test may say greater than 250, and a more recent test say greater than 2500. We discussed this. The sensitivity changed. Any value reported under 250 are consistent between the tests. Okay, excuse me again. Am I protected from COVID? We can't answer that because nobody knows for sure what the numbers mean. And so we can't tell you. Are the tests ordered by my doctor the same as LLS? From my experience, the answer to that is no. That doctors order tests and they're processed in their own academic centers or their own lab, or they're sent to Quest, or they're sent to many different labs, and they are not apples to apples. So it's difficult for us to compare. Okay, what if I've been off treatment for a year? What does your study results say about my antibody response? So we haven't broken this down yet. As I've said, the study shows that the longer off treatment, the better. And anti-CD20 therapies such as Rituxan or obinutuzumab appear very long lasting. And I mean probably a year long lasting. And that leads to the question, should I hold up or stop my treatment to receive a COVID vaccine. Of course, this is a discussion to have with your doctor. And since we do not understand the time frame between no treatment and vaccine response, we think it's more important to continue treatment and keep your guard up. Personally, I've told you that I have CLL and I've had CAR T therapy, which wipes out my B cells. And at the moment, my CAR cells are still working and my antibody response is 0. Now, frankly, between all of us in this room, I would rather be cancer-free and wear a mask and be careful than stop my treatment just so the vaccine can become effective. That's my editorial comment. Take it as that, as a personal comment. What activities are safe for me if I did show some antibody response? Again, we can't really say. You know, we recommend keeping your guard up, wearing a mask, social distancing, and hand washing. In my case, again, knowing what I just, shared with you, my approach is that, frankly, I don't do indoor dining. I don't go to a movie. I don't go bowling. I will meet socially with friends in an outdoor environment. And, I have children and we have very close friends. And if we know where they've been and how they act themselves, we will see them on an indoor social basis, but otherwise very careful. Okay, do I need a booster shot as a blood cancer patient? Frankly, this is yet to be determined, and that's what we are studying. How can I get a booster shot? Actually, we're not calling them boosters anymore. We're calling them third vaccines, because we say third because they haven't been approved yet for Johnson & Johnson. They've just been approved for the mRNA vaccines. And basically, for blood cancer patients between now and September 20th, as I did, you can go to most any location and you self-attest that you are immune compromised and you'll get a vaccine. The recommendations are to receive the same type mRNA as the initial doses. Having said that, if you had a Pfizer or Moderna and you can't find the same kind of vaccine, you can mix and match. We have a very small number of patients who it appears that Moderna may create a better response than the Pfizer vaccines. And we see this based on the number of blood cancer patients who are seronegative. Now, we don't quite understand why, although we know that the Moderna vaccine packages more mRNA into its vaccine than the Pfizer group does. So that may be a clue. And just to, say that, how can I enroll in clinical trial for boosters? This is the number for the Montefiore Medical Center for those of you who live in the New York area, if you want to join their study. The reason for this study and these kind of studies are important because they're not only giving a third dose, but they are monitoring for safety and they're doing blood tests on a periodic basis as a clinical trial would do. For people who are in our registry, we are only asking them if they've had a third dose. We are checking an antibody response, and that's it. So it's an observational study, not as tight as a true clinical trial. We are accepting current registry participants for follow-up testings. And I say here, when the formal booster study begins, we will promote it. But frankly, as it looks now, we're just going to accept all comers, and there will really be no formal third dose study. We are monitoring these studies that are available. Are the side effects from the vaccine worse in blood cancer patients? The answer is no. Our side effect profiles, and we have survey results from 3,500 patients, show us that their side effects were the same as normal healthy individuals, meaning number one side effect is a sore arm and so on. Does the vaccination lead to relapses of blood cancer? As best as we can tell, and in current studies that we're monitoring, there are no reports of this. There are coincidental reports to us of people who say, by the way, I had a vaccine and now I'm doing worse with my blood cancer. But we cannot say anything that that's related to the vaccine. There is a question among people who do not want to get the vaccine. Was the speed of the development, did that mean that steps were missed? And quite frankly, the answer is no. These vaccines were developed over years and decades. The reason they were so rapidly approved is because when they each onboarded 30 to 40,000 people, COVID was running rampant. And the speed of these participants who were either in the placebo group or the vaccinated group who were exposed to COVID and became infected with COVID happened very rapidly. So they were able to tell how the vaccines worked. And that was the step that was accelerated. And that's because of the statistics. When will the FDA fully approve the vaccines? Well, frankly, we don't know. We hear rumors that it's going to come soon, and we do believe that. Based on treatments that a patient may receive, should vaccines be postponed? And frankly, the answer is no, because even though you may not have an antibody response, as I said, we're studying T-cell responses, and we think everybody should be vaccinated. Now, having said that, if there's a patient who's currently getting bone marrow transplant treatment or something like that, then of course they're not going to be vaccinated until their marrow is ready to respond. So I want to give that as a qualifying note. Bottom line is get vaccinated, act unvaccinated. For additional questions, you can contact us at our email address, which really is best, at pact@LLS.org. I'm sorry, I see a typo. It says comma, but it's LLS.org. We have a voicemail box at this number. I hate to say it, nobody picks up the phone. It's just a voicemail box, and we respond faster to emails. If you have other general questions, about blood cancers or trials or that kind of thing, then please call our LLS Informational Resource Center at 800-955-4572. So these are our contact information items. This is a long URL, I'm sorry. You could visit LLS.org. You can search for COVID, and we have a lot of pages of FAQs and information on COVID. Please stay tuned for future updates. We actually today submitted a paper again to Cancer Cell on our first analysis of 30 patients who did receive a third vaccine and how they fared. I cannot report on that because, as you might understand, when we submit an article, the data is embargoed until the research journal says yes or no. So, but we are hoping that it'll be accepted soon, very soon, and then we can post that data. I really thank you for your participation and your attention, and I'll stop sharing my screen, and I will look forward to your questions. So thank you very much.
Thank you so much, Dr. Saltzman. That was extraordinary, absolutely phenomenal, and so clear. I'm so grateful for the crystal clearness of everything that you just shared. I learned so much from you, and I'm grateful for the WM data that's so hot off the press there that you were able to offer us from just days or moments ago. Thank you for doing that. That was so eye-opening. And some of the things that I'm going to take away when people call a little bit wigged out, like you say, about antibodies that are under 0.40, that they can't be evaluated or measured by that test. That's a really critical point for folks to understand, thinking they have zero immunity or no immunity. The same way, just not knowing whether, do we have, do we have immunity? What does it mean? not being able to drill down and know the absolute answer to that causes so much anxiety, because we have been living, with this lockdown and with such caution for so long, that the idea that this continues to be endless, even though there is a vaccine in our arms, and even though there's a second vaccine, and even though there'll be a third one, that we have to continue living with this, tolerating a high level of ambiguity. And thank you for the clarity in that. It came across as hopeful as well. So it's a delicate balance to know how to do that. But you struck that chord, and I wanted to thank you for that. And the 250 to 2,500 change, not having to do advanced mathematics to figure out what that 250 actually means, but to understand that 250 still means 250. And we don't have to do anything statistical to that number is critical. I know that will answer a lot of people's questions. And the lymphomas, those results that you put up comparatively, disease by disease, that was, for me, that was a really key takeaway. And the WM, in terms of the 74% that responded, boy, is that ever a different number than it feels like when we're talking informally as a community. So on our online communities, or the buzz on the street, or the people who are calling Lifeline, it just feels like there's such a low response. And I believe that your ibrutinib and IVIG, the information about what could skew our response, what kinds of treatments we might have been on in the past year that could make it the number be, a more disappointing number than many would want, might be the case. Also, I'm thinking, Dr. Saltzman, that people who are on, I'm curious what your thoughts would be, people who are on the online community might be people who are in treatment or actively, seeking information because they are already experiencing IVIG or this and that. So that's why it gets a little skewed in terms of who's reporting on, hey, we don't have any immunity. The ones who are not in treatment might have, are gone fishing.
Right. So I'm not focused at the moment on the LLS community itself. This is more our registry. And I do need to admit that our study is biased. I mean, we are biased to people who have internet access. We're biased to people who understand how to use a computer.
Right.
We're biased to people who can accept an email with a lab slip attachment and download it and print it and find a lab and go. So this is a biased study, and for what that's worth.
I appreciate you sharing that. I'm wondering, there are 402 WMers in your LLS registry right now. I am proud to be one of them. Should we continue to plug that registry so that we get more WMers in there, and we want to get the word out, and explain also to people about going to LabCorp to get tested, so that you will have results of the 250 missing folks. What do you think about that? Too late?
Bottom line is we would appreciate every person being part of our registry and sharing their medical records, because ultimately the antibody testing is something LLS is doing as a service, as a study, and to help a person's personal curiosity to know where they land. And so that's something we're investing in at the moment, right? This is in the present. Down the road, we really want to invest in medical records and treatments, and not only understanding the records from a hematologist perspective or an oncologist perspective, but you can look at me and you can see that I'm not a younger fella. I'm a little older, and so because I'm a little older, and frankly because I have CLL, I'm prone to skin cancers, as most CLL patients are. So I have shared the records of my dermatologist because I've had basal cell carcinomas excised from my skin and squamous cell carcinomas, and I want that to be part of our medical record research. I see an internist. I want those records to be part of it. I see a urologist because I'm a little bit of an older guy, and I want those records to be a part of it. And as we look at medical records, not only are we looking at the outcomes of treatments for blood cancers, we also want to link those to what I referred to as comorbidities. Do people with hypertension or diabetes or rheumatoid arthritis or Crohn's and colitis or benign prostatic hypertrophy or any, of the things that people might have, are they a factor in how blood cancers are treated or the results. And the only way we're going to see that is to compile records so we have enough of them to see trends. So that's what we had. Joel keeps raising his hand and I keep looking at it on my screen. So you have some burning question. Why don't you do it? I know Lisa's doing the questions, but go ahead.
Okay,
You're
I'm
unmuted.
unmuted.
Here you are.
I am one of the 152 WMers who gave blood, and I was treated 6 years ago, so that's not a factor. I was one of the 74% who responded to the vaccine. I was one of the 59 on no therapy. My question is, why didn't I get an invitation for the Tcell study? Although, since you're not giving results, I don't know if I'm offended. Probably not. I mean,
Okay.
and the other thing is why, when will you invite people to the booster study? Because I'd like to get a booster as soon as I can.
Right.
And
So-
thank you for the information that we only have to self-declare. We don't have to prove to
Right.
the center that we're immunocompromised.
Okay, so let me answer those questions because that's fine. So the T-cell study, how do I say this politically correctly? LLS does not have the funds to measure T cells on every person who is part of this registry. It's a very expensive test. And we're looking primarily to answer the question of people who did not register antibodies. Do they have T cells? So majority of the people invited were people who were on the seronegative side of the pendulum. Now,
I see.
for the other control arm, we randomly invited 400 people of various blood cancers, Waldenstroms, CLL, Hodgkin's, so that we would have a control arm to look at people who had CLL and no response and CLL and had a response, right? Now, of the 1,100 people we've invited, so far, 500 people have had their blood drawn. And we're keeping this open until the end of August for people to have their blood drawn because it takes 6 to 8 weeks to get the results. And we don't want this to go on forever. Now, once we get to the end of August and we then evaluate how many people of what serology and what blood types had their blood drawn, we may invite more people. Because Joel, if nobody with Waldenstroms who had a response went and had their blood drawn and we need some people in that control arm, you may get an invitation. But I can't guarantee that right now. It's just the way we're handling it. As far as the third vaccines, the way we're looking at it, a week ago, I would have said that as per an email that we sent out, we're inviting you to a booster study and let us know and all that kind of stuff. Everything's changed in the week. Everything has changed. And now, like I said, essentially anybody can get a third dose. And so we will be offering, and we'll send out another notice that anybody who's going to have a third dose will be able to have their antibodies checked. And it will be a 3 to 5 week time period from the time of the third dose. If you get it too soon, it doesn't register. And if you get it too late, it's not as good. So there's a sweet spot. So we are in an avalanche of people who, even before we sent out our note on Monday, they went this weekend and had their shots. And frankly, we would prefer that if people have not had an antibody test within the last 3 months, that we get a test before their third vaccine and then after their third vaccine, so we can measure if the antibody response has degraded over time and what the booster will do for it. Having said that, people are just rushing out and getting it, and we cannot keep up with the requests for lab slips from thousands of people who say, I'm going to get it this afternoon, send me a lab slip. We just don't have the staff and we're not automated enough to do that. So I apologize for that. But for those who can bear with us, we're going to try and do that. And for those who just want to get a booster, I call it a booster. Now we're going to call it a third vaccine. Please just go and get it and send us an email that I've received it, and we will send you a lab slip. I mean, that's just as I can't tell you how fluid things are and how they've changed almost from hour to hour. And I apologize for it, but we are just trying to keep up with what's happening. So I hope I answered your questions. Back to Lisa, who's managing the FAQ line. Thank you, Joel.
Thank you, Dr. Saltzman, for jumping in there. I really want to thank you also for drilling down and giving us your Dr. Larry's approach, because a lot of folks want to know some specific guidelines, and the ones that you're describing are the ones that I'm living by as well. And so it was just nice to hear you kind of giving concrete examples and giving people some goalposts so they can figure out where, together with their healthcare provider, where they feel safe and what's the best decision for them. But the get vaccinated, act unvaccinated, I want a t-shirt that says that, because that is just the best logo I have ever heard. And I'm just gonna go with that. I would love to get to our 10 pre-submitted questions and ask folks, instead of raising your hand or doing the, the hokey pokey, go ahead and put in the chat your question. After we finish the 10 questions that were pre-submitted, we'll go ahead, jump into the chat and try to get to those. But I'm going to do, I'm going to do chairperson's preference. We've got Pete Denardis with the cutest baby on the planet on his lap right now, and he threw a question in the chat, and because of that adorable baby and his amazingness, would you like to just, Pete, ask your question first? Was there any analysis of response in folks with hypogammaglobulinemia, or would that just be the same as looking at the data with regards to those getting IVIG? So a little bit of a specific nuanced question here before we do our pre-submitteds.
Yeah, so at the moment, the answer to that question is no, because we didn't ask that specifically in our survey. So the answer to that question will be coming once we crack open the medical records. And that's happening right now, but it's a very slow process to robotically extract data out of records and then manually, with a clinical staff, review them to make sure we have everything. LLS has created for this project what's called a data dictionary and a data ontology set of thousands of data points, diagnosis, treatments, medications, and the like, that we're going to be extracting. So your question, big word, hypogammaglobulinemia, is one of those data elements that we'll be looking for, and then we'll be able to report on it. So the answer currently is no, and the answer, I hope, will be yes when we get into that.
I think we get a big thumbs up from the baby. Right. Okay. Thanks, Pete. It's good to see you, Pete. Thanks for hopping on and bringing cuteness with you. Okay, the first of our 10 questions, Dr. Saltzman. Were healthy control subjects included in this study? And if so, what was their mean antibody response?
Okay. So in our study, we were looking only for blood cancer patients. Now, do we consider blood cancer patients not on treatment as healthy? Wait and watch. Are they healthy? They have blood cancer. I don't know. I assume that every person with a blood cancer has a bit of a compromised immune system. So I would say that we did not invite any healthy volunteers to our study. And the data numbers that I reported on the 200, 130, those serology numbers, those were based on healthy volunteers from the clinical trials of the vaccines. They are not in our registry.
Okay, very helpful. And we're wondering what long-term follow-up is planned. So if I'm in your, I sent in my lab course, slip, and I got my slip, I got my blood work done, I got my results. What should I expect to hear from you? Is this an ongoing relationship, or is it one and done?
No. So we are planning that we will be monitoring people for up to a year, after their, well, what would have been their, final vaccine, be it one and done J&J or 2 of mRNA vaccines. Now, knowing that Americans will be offered a 3rd shot come this September, everybody, whether you're blood cancer or not, we are looking at extending our program to include a year after the 3rd vaccine. So if you get a 3rd vaccine now or September or October, you can figure you'll hear from us somewhere 6 months after that 3rd vaccine or 12 months after that 3rd vaccine to check again. So we're in the process of amending, we have to amend our consent to allow people for further contact and for perhaps additional blood draws than we had planned originally. So we are actually in the process of doing that right now.
How interesting. Okay, I will wait to hear from you guys.
Mhm.
Do you think the isotype of the response matters? So WM patients are often low in IgG and IgA and may only make IgM antibodies. Does this make a difference?
So I understand that. And again, I sound like the little broken record. We will know more when we crack medical records, and we're assuming most doctors, check levels of IgM and all these antibodies on a regular basis in blood cancer patients, especially Waldenstroms. And we hope to be able to make that correlation. So stay tuned.
Okay, will do. Here's a little bit of a crystal ball question. Is there any data on whether we are maintaining our semi-quant levels? All of us want to know. Is time running out?
Well, that's a very good question, and I'll answer it on 2 levels. One level is we'll look at the Israelis, which are probably the most studied country in the world based on their health system and access to the Pfizer vaccine. And the reason they have now lowered the age to 50 years old to give a 3rd vaccine 5 months or 6 months after their final, what would have been the pairing of 2, is because they're seeing the antibody levels wane and they're seeing infections with the Delta variant on the upswing. So from studies there and from studies in the UK, yes, we're seeing antibodies level decrease. Having said that, again, I'm just going to be, a little nuts about this. Antibody levels after a vaccine always fall. The body does not keep itself chock-loaded with antibodies for every cold virus we've encountered or every vaccination we've had. We have memory cells that float around, and then if they see this entity again, all of a sudden they wake up, their T cells wake up, their B cells wake up, and they go to fight it. But I think if you ask a researcher, which I am not, we would expect every antibody level from everything we've ever been exposed to go down. The problem currently is that the COVID-19, as we call it, infection and the variants are not going away. And we need to keep fighting these. And so people are intensely looking to see what's the level that we need to make sure we're protected. Nobody knows. And so that's the long answer to that short question.
Oh, that was a great answer, though. You just have a way of getting right to the heart of the matter. You don't mind coming with me to every single medical appointment, do you? Like, that would be okay with you, just to explain it all the way you do so beautifully, and all of ours. Here's a question from New Zealand. So shout out to Lee and our New Zealand folks. Are there clear guidelines for measuring antibody immune response using ELISA, E-L-I-S-A? In New Zealand, we're told there is no clear measurement. Is that so?
So it's very interesting. We decided there are many academic centers using ELISA testing to look at T cells. It's an older technology, and again, I may get this wrong, but from when we were looking at this, it required a blood draw. If you think about a tube of blood as 5 mLs, that's a big tube of blood. it would take a blood draw of 12 tubes of blood to get enough blood to do ELISA testing on T cells. And so we at LLS decided that that would be too much. I mean, I would, if I had to go to a lab and they're drawing 12 tubes of blood, I would just be, you know, again, totally wigged out, which is why we went to this research only T cell genetic test, because that requires 3 mLs of whole blood. It's 1 tube, and it's a research test. So we're not doing ELISA tests. A lot of academic centers are, and I don't have the reports of those. So I really don't have an answer to this one.
Okay, so then I'm going to ask you another thank you for that. I hope that was helpful for our New Zealand folks. And I have another crystal ball question for you. And then we're getting to the end of these, and we're going to hop into the chat, and Meg and Marla, we're going to grab all of your questions that are there. What's the latest impressions of the breakthrough infection rate for fully vaccinated folks? The $1 million question here. We're really asking you the tough ones.
It really would be a mess to do this, but I just saw a posting. If you know, hang on a minute, okay? Can you hang on a minute? I may
we have
be able
all
to,
the time in the world. Are you kidding?
I may
Please.
be able to actually.
Please, because I'm going to hop into the chat now. Take this opportunity because we only, please do your thing.
Hang on a minute, and
fabulous.
Let me see if I can get this up. I have something to show you.
Oh, brilliant. We love it.
Boy, but where is it? I'm sorry. Just okay.
Wait. You can hop off for a sec. Yeah, no, or we can just do, I actually wanted to ask someone a clarifying question.
Here,
you got it? Ooh, that was fast.
Yeah, I got it. So let me share my screen, okay?
That was exciting. Yes, please.
Yeah, that's exciting. Hang on. But I don't know if this will answer your question exactly, but let me do this. Okay. So I just got this today. And can you see my email box?
Perfectly.
Okay?
Yep.
And this graph is breakthrough COVID infections and deaths by state.
Wow.
Now, this was just reported, and it's not blood cancer patients. So this is total people. There's no data on breakthrough infections in blood cancer patients. And let me say, as you're looking at this, that the CDC stopped collecting data on breakthrough infections unless people were hospitalized. So we don't know who's having a breakthrough and they're just having a little cold and they go to their doctor and they take some Tylenol or Claritin or Benadryl, you know, or whatever nose spray to help themselves. We have no idea. So these are the breakthrough infections, hospitalizations, and deaths in different states. So where I live in California, the percent of COVID hospitalizations and breakthroughs is 0.4%. Deaths are 0.5%. What's the biggest state? Let's see. I'm going to scan it. Arkansas is not very good. 4% of patients, all hospitalizations are breakthroughs. That's not so good, is it? And, you know, if you look at most of these, they're all like in the, 1%. Rhode Island is 4%, death rate of 1.4%. Utah is not very good. 4.3% hospitalizations are due to breakthroughs. 2% of the COVID deaths are due to breakthroughs. Wow. Vermont looks very bad. 5% of deaths are due to breakthrough infections. Anyway, so this is a current table, and I'm not sure where it came from, but it just popped in today. So
that's incredible.
That's an answer to your question. Lisa, I can send this to you.
Oh, that would be wonderful, because then we can send it out for folks.
If you remind me, I'll send it to you. Okay.
Thank you kindly. I will. Breakthrough data. Thank you. That's fascinating. If it's okay to send it out together with your slide deck and this. Oh, terrific. A couple of weeks ago, someone writes they had a 3-month post blood draw for antibodies. Is this data still coming in for the group?
Yes. And
Is that-
so we are actively looking at the, like somebody asked, the waning serology in 3-month group. And actually today or tomorrow, we're sending out another 400 invites for people who include a group who had vaccines now 6 months ago. They've only in our study had one blood test, which is more than 3 months ago. And so we're inviting them, and we're inviting people who had vaccinations more than 3 months ago and blood tests that have to be more than 3 months ago. In other words, if somebody had a vaccination 5 months ago, but through our study, they had a blood test 2 months ago or 6 weeks ago, we're really not, I mean, you may be interested, but we're not interested yet in doing serologies every month or every 6 weeks. You know, we have to give it some time. So yes, we're looking at that. And yes, we will publish that when we get enough data to look at, which hopefully is coming soon. We felt that right now, frankly, the paper that we've just submitted on people who've got their third vaccines was more important and more timely right now than, quote, the 3-month serology, frankly, because as I said, that data is coming out of other countries. So
Understood.
there you go.
Well, when we send this link out, we can send it with a push to really ask people to join, to ask those who have already joined the registry to be sure to request their lab slip for LabCorp. And just we'll do our part, because we really cherish this partnership so much with LLS, and we, you know, at the IWMF want to support your efforts as much as possible. It's all of our best interests. So we have about 6 questions in the chat box. Dr. Saltzman, how are you doing? Are we okay for time? If we will get you out of here in advance of 4, we promise.
Yes,
we
we're
want
good.
to. Oh, fantastic. Thank you so much. So Meg Mangan, who is the moderator of our private Facebook group, asks, do side effects or the absence of side effects following a vaccination correlate with an immune system response? A really hot question. Many, many people wanted to know. You spoke about the fever that you had. I've had people call and say, I didn't have it. I had it really badly. What does it mean?
So the true answer is, I'm not sure anybody knows. I wish it did, because when I had my second vaccine some months ago, I actually had a severe reaction with 4 days of fever and a temperature up to 102.5 that lasted way too long. And I thought, goody, you know, it's working. And then I got my serology and I had no antibodies. So really, personally, I can't say, and I don't think anybody can say that there's a correlation between the cellular immune response and what the antibody levels are. That's the problem, right? That's our problem.
But we have to accept that goes along with the tolerating a high level of ambiguity and just getting vaccinated, acting unvaccinated, and staying in the safe zone. So I appreciate the consistency of your message there. We have another question from Marla Chow, who's our hardworking support group leader in Southern California. Hey, Marla, who asks, what about getting a 4th vaccine if you didn't respond to a 3rd? She's already at the 4th. So I love the way how quickly we mobilize.
Here's the problem, frankly. And the reason we want to support academic centers who are truly doing clinical trials on additional doses of vaccine is because we have no safety profile on getting these additional vaccines. And we have no profile on what happens if a person keeps challenging themselves with vaccine. Personally, I'm concerned. We have one patient who wrote us who keeps going, and that person now has had their 5th vaccine. How they get it and how they qualify and who they have to tell they have not yet had a vaccine and they get, I don't know how it's done, frankly, but I'm very concerned that we need some safety data and some response data before people go out and do this. Now, you know, by the by, wink and a nod, you know, if people write to us and say, we're part of your study and we had our 4th dose, can we check our antibodies? You know, we'll probably say yes. LLS is funding this. This is not a free thing. LabCorp charges us for every test we do, and it's not inconsequential. But I just really wish that if people wanted to have their, okay, now the CDC, FDA has looked at vaccine profiles, at least from Pfizer and Moderna, and said, okay, a third dose is okay. So we have to go by that. Having said that, we haven't studied it in our population. If people want to go for more doses than that, I really wish they would find a clinical trial and be monitored by their physician, not just LLS doing an antibody test. I just can't get my head around that, frankly.
Thank you for that cautionary response. I appreciate that and hope people will take those words to heart. Sherry Ann Hall, who is our support group leader in Ohio, is asking any word of the second blood draw, like a 3-month follow-up study I had drawn in mid-July. I think you might have answered this. Do you feel, is there any more that you would want to add to this, or do you feel like that was sufficient?
No, I just think there's a little confusion about people who we know when they read our study and they read our protocol and they mark their calendar and they say, we're going to have a blood test, after the first, after the second, 3 months, 6 months, 12 months, whatever, that there is a little bit of ambiguity in that. And we should have been clearer that we are looking for studies down the road, but we really don't want any blood test that is within 3 months of each other. If that makes any sense. And so, that's the ambiguity. And so when people are writing us for their lab slip because they say, well, it's been 3 months since my vaccine, if they've had a blood test within 6 weeks or a month, we're really saying, hang on a while, because we're trying to manage this in a little bit different kind of way.
Okay, thank you. Bill Block from Seattle, our support group leader there, asks, I share the question about whether to do a fourth if I did not respond to a third. So he had two Pfizers.
I think that's already answered. You can skip me.
Yeah? Yeah? Okay,
Yeah.
no antibodies. And then you had a Johnson & Johnson. I just think it's kind of interesting what you did. And then again, no antibodies. Would a fourth make sense? Okay. Interesting trajectory. Yes, thank you, Bill. You're in the interest of time, Bill. Meg Mangan, you mentioned September 20th. Is that the last date that third shots will be available to immunocompromised patients? We heard I think there might be some.
I think the answer there is that at the moment, the third shots are approved for immune compromised individuals, whether they have blood cancer, colitis, Crohn's, MS, rheumatoid. I mean, there's millions of people in that category. On or about September 20th, whenever the federal government approves this, then that distinction goes away. And anybody, any adult, will be able to go in and get a third vaccine. So, I think from my perspective, all I can say is that in the very beginning, last December, January, February, it was very hard to make appointments for vaccine. The supply was limited and we were all scrambling. Right now, the supply is better, although I wish it would be used for unvaccinated people to get their vaccines, but the supply, I'm sorry, that's an editorial comment, I apologize. But the vaccines are more plentiful. And I do think if you're immune compromised, we have a window to get the vaccines now before the horde knocks down the doors of the pharmacies and the medical centers. So I think we have a period of time where it would be wise, if we want to do that, to go ahead and do it right now while that door is open and the supply is there.
Okay, that's very helpful. I'm going to admit that I texted my doctor in the middle of this talk. I'm just gonna go ahead and fess up. I actually did it. Okay. When am I getting on the calendar? So here we have an interesting question from Mel Horowitz in New York. Mel is a longtime support group leader and on Lifeline, he leads one of our fun categories, which is living well and thriving with WM over the 20 years club for over 20 years. And he asks, my original COVID vaccine was J&J. Can my next vaccine, my next booster, be Pfizer or Moderna? What do you do now?
So,
Great
so
question.
I'm not expert in this, and I'm trying to keep up. I do know that the government has not yet approved a J&J second shot. I have heard anecdotally that people who've had J&J can find themselves in a position to get an mRNA vaccine. And I think that we'd be happy to study that. That's all I can say. I mean, if somebody finds their way to that, then we're going to want to know who's mixing and matching and what the results are. I mean, I just think that's a natural outgrowth of what we're doing. I cannot comment on the safety of doing this. I cannot comment on how a person would go about doing this. But I think we'd want to know. We'd want to follow it.
Thank you. Sherry Ann Hall asks, what happens if your treatment status has changed? So if you're in...
Well, I guess I would, I mean, your treatment status can go from one to another. You could either be starting treatment and you've already had vaccinations, in which case, I don't think there's any difference in what you do. If you start treatment and you're due for a third shot, go get the third shot. Mm-hmm. If you're off treatment, then I guess the question would be, okay, now I'm off treatment and I should have a blood test that tells me where I am now or where I was before. And then I suppose talk to your doctor about when would be a good time to get a third shot or another shot. I mean, I've already said it, that there are certain treatments that are longer lasting than other treatments. So frankly, if I was on an anti-CD20 treatment, you know, rituximab or obinutuzumab, and I just stopped, would I want to get my third dose the day, the week, or the month after? You know, I'd probably say, probably not. It's not enough time to wait. I mean, I think that's what people are seeing. But, you know, will it hurt me? Probably not. You know, I mean, it just, these are really discussions that need to be held with, not with LLS. We're not a medical provider. We can report statistics, but this needs to be held with your doctor. And I want to keep saying that, although I know the doctors don't have the answers either. So this is, you know, a problem, and everybody's trying to roll this out as fast as they can. So.
Exactly. So here's a question for LLS, and these are the last few questions, so we'll be wrapping up soon. Can you explain what is meant by a cellular immune response? Definition?
Well, I think I implied that, which is, you know, the body remembers what it's seen before. And so, like I said, if you get a sore arm from a vaccine, or a fever from the second vaccine, or some symptoms, that's a cellular response. That's the body's response to something foreign invading the body. That's a cellular response. And so, you know, most people even get that. If I get a flu shot, frankly, I get a flu shot every year, and every year I get a sore arm. That's a cellular response. I think that's the example that we can use.
Very helpful. And will so-called booster shots for healthy people be half the dose of the primary series? Like, what's in that booster that we're getting?
So what we're seeing is the doses that will be given are exactly the same as the doses that were given for the mRNA vaccines first and second times. It's exactly the same dose. It's exactly the same vaccine. It is not based on the variant yet. It's exactly the same vaccine, same dose.
Okay.
The only doses they are trying to figure out best dosing for are for children under 12, and especially children under 5, because an adult dose of a vaccine may not be the right dose for a child. So that's why those trials are going on now to validate the correct dose for children. So at some point in the future, we'll get the okie dokie to get every person vaccinated, which right now, as you know, we don't. It's only children over 12. So that is all I know on that.
Well, here's a last question for you, Dr. Saltzman.
Yes.
And I see that the sun rose in New Zealand. Outside of Lee's window, it was dark at 6 a.m. when the meeting began, and now there was a beautiful sunrise and lovely morning light there. So wonderful hopefulness here. We asked this same question of every Rockstar doc who meets with our support group leaders. And we wanted to ask it of you. What is the one thing that you wish every WMer or every blood cancer patient, if you prefer, knew today? So is there one thing, one message? Because every support group leader and Lifeline person and volunteer international affiliate on this call represents, and everybody who's going to be listening at home to the recording, represents so many people that they can share the word with, and their support groups that they can spread all of this good information. So what's the one key takeaway that you would want them to leave with?
So I assume you're asking about SARS-CoV-2, right? Not just about life in general.
Oh, heck, answer any way you like.
Yeah.
You could give
So,
two answers, one specific to COVID and one about life.
Okay, two answers. So as far as SARS-CoV-2, my answer would be, it's more devastating in blood cancer patients than people recognize. And it shouldn't be taken lightly. And whatever is recommended for the normal, healthy populations should be looked askance by blood cancer patients. So I just think we should be more careful and more hesitant than what the recommendations are. It was no surprise to me that in June, when the recommendations were, if you've been vaccinated, go have a good time with your life, and they didn't include immune-compromised patients in a disclaimer, it was no surprise to me that this now increase in events and infections was going to happen. And I just think that we should all be very careful. Now, answering the question in a different way, I wish that everybody who has a diagnosis of blood cancer does not get down on themselves or their families, spooling into despair that I have cancer and oh, woe is me. Because I know through LLS, I'll just give them a shout out. Every treatment I've been on has been researched by them over the last 20 years, and I'm alive because of them. And I was able to travel a lot in the first 10 years of my disease, even though I was treated with everything under the earth. And, I think that people should be more optimistic than they might want to be. Now, I admit that given COVID, I am not traveling anywhere, right? We had a trip planned to Hawaii in September, and we booked it many months ago thinking by September, everything would be good. And now that we're looking at it, no way am I going. So, those are my two answers. And I really appreciate your time and attention. And to everyone out there, thank you very much for supporting us. And your foundation is great. And thank you very much.
Thank you, Dr. Saltzman, from the bottom of my heart for that fabulous presentation and conversation. I learned so much that was so helpful, and I know it's going to be so helpful to WMers and blood cancer patients across the world. We're so grateful for all that you do personally with such passion, and for the work that LLS does every single day to help improve the lives of all of us blood cancer patients. As LLS says, it's in our blood, so we are all family, and we are all very grateful to you, Dr. Saltzman. Thank you so much, and I wish you much aloha. Even though you won't be in Hawaii in September, I hope it comes very quickly and you will be on the beach soon enough. I want to be sure that everybody knows that the IWMF now has specialty support groups for WMers. And if you know someone with Bing Neal or peripheral neuropathy, or perhaps you've met a young WMer or a WMer interested in meeting other people of color who share their diagnosis, we have got a group for you. Please contact the IWMF office by phone, email, or snail mail, and we will connect you right up. And lastly, registration is now open for our virtual Ed Forum in October 2021. Be a part of the magic and imagine together with us a world without WM at IWMF.com. Dr. Saltzman, thank you again so very much. We're so tremendously grateful. Thank you, thank you. And we're out, folks. Shelley, you can stop